So welcome everyone to this session. Welcome to the festival tomorrow if this is your first session you're joining, and welcome back if you've been in previous sessions. I hope you've been enjoying the exhibitor booths as well. I'm just going to introduce this and then I'll lead you to it. I'd like to thank the Wasdell Group, one of our partners who've been supporting during the pandemic, based right here in Swindon, and also want a quick shout out to Zeiss as well, who are giving us a quite unique opportunity to get remote access to the labs for this event. If you do have questions, try to put them in the Hoover Q&A. You'll see there underneath the window, rather than within the Q&A of Zoom, which is embedded within, and we'll get to those as we go through. So I'll just introduce Roland Pease. Roland Pease is the regular presenter on Science in Action on the BBC World Service, a programme that's been broadcast across the globe, the latest in science every week since the 1960s, but I don't think Roland's been doing it since then. For the past year, much of the programme has been devoted to exploring the science underlying COVID-19. Brought up near Wantage, Roland has lived in Swindon area since 2007, so he's local to us. And so over to you, Roland.
And we have got a fabulous list of people coming up to tell you all kinds of amazing science that's been going on in the UK about coronavirus. Really in the past year. I'm going to hand over very quickly, though, to Marty Jopson, who's going to introduce us to the virus itself. We've actually got a live feed from an electron microscope. Marty, take it away.
Thank you very much, Roland. I'm still. Oh, am I on? Yes. Am I on? Am I on? Yes. So my name is Marty Jopson. I'm a science communicator, and so basically during lockdown, rather than going out and talking to people about microscopy, I actually sat here at my desk and sorted out with Zeiss how we could drive microscopes remotely. So what we've set up with the lovely people at Zeiss is there is this microscope here. I just asked Steve, who is down in Cambridge driving it while I'm up in Yorkshire, I just asked him how much it was worth. He said £350,000. Don't break it, he says. I'm not even near it. I can't possibly break it. So anyway, let's go and have a look at what you can see down this microscope. This is what you can see if you peer down one of these microscopes. And what we've got under this microscope at the moment, at least, and this is an electron microscope. It's a scanning electron microscope technically, but we're using it as if it was a transmission electron microscope. It's all very complicated. But the point is, what you're seeing there is a little grid, a gold grid, with a layer of carbon over it, and then virus particles scattered all over it. Now, this came from Matthew Hanna at Public Health England, so thank you to them. And in terms of scale, if you look at the bottom down here, at the very bottom, there's a little scale bar. And at the moment, it says 100 micrometers. So this is sort of a micro, a mu. So a micrometer is 1,000th of a millimeter. So that means it's 0.1 millimeter, 1/10 of a millimeter, that bar. And that is conveniently the size or the diameter of a human hair. So we're already zoomed in quite a lot, but we're going to have to go in a lot further than that. So I'm going to zoom in now. And all I have to do is click over here on the, well, you're supposed to call it magnification, increase the magnification, but I just call it zoom. So we're going to zoom in. Each one of these little squares, by the way, is about 35 thousandths of a millimeter. I'm going to zoom in a bit more, keep going, and you can now start to see sort of the speckledy nature of what's between the grids. Now, I'm going to go in a bit more, and now if you look at the scale bar now is showing 10 micrometers, so that's 10 thousandths of a millimeter, that size. That bar at the bottom now is about the size of a blood cell, a red blood cell. So still no real sign of virus particles. These virus particles, by the way, they came from a patient, a British patient. We don't know who they came from. Even if we did, we probably couldn't actually tell you who they came from for confidentiality reasons. But let's keep going in a little bit further. So we'll go in a bit more. Now, the original, the first people to actually see coronavirus was a lady called June Almeida in 1967, when they were just first started messing around with electron microscopes. And they managed to identify and isolate a coronavirus. It wasn't obviously the coronavirus that's giving us so much trouble at the moment. So we'll keep going in. And now you can start to see all of these little specks, okay? And these are coronavirus particles. And some of you might be noticing over here, for example, there's a sort of a rather obvious square artifact. And that's because this is an area we've looked at before, and you actually burn out what's on the actual sample when you look at it. So there's a certain amount of sort of destructive nature to what we're doing. But anyway, let's keep going in. Let's keep going in. The scale bar now is 4 microns, which means that a blood cell would fill about half the screen. Keep going in. We're at 13,000 times magnification. Okay. And now we've just moved into the nanometer range. So a nanometer is a millionth of a millimeter. Okay, so a billionth of a meter. The numbers start to get ridiculously small, and we'll keep going in, and then I will center it on this specimen here. And we'll go in once more just to really zoom in on that. Look at that. And there it is. That is a coronavirus. In fact, there's 3 coronaviruses, one next to each other. There's one here, obviously, and one here and one here. And this one in particular, Steve had a look at this. Steve is the genius that's doing all the real hard work here. He's the one that sets all this up down in Cambridge. I'm sitting up here. I just drive the microscope remotely. But you can see all the little specks around it that give it the name coronavirus. That's why it's called a coronavirus, because of those specks. And those are obviously the spike proteins that we hear so much about on the news. So there it is. There's a coronavirus. There's the scale bar at the bottom, 100 nanometers. So this coronavirus is what? They're about 60 nanometers across. They weigh about a femtogram. I'm blathering on. I'm not sure if and when you want me to stop talking, Roland.
I was going to say a femtogram that people may have seen just last week. Marty and I had been talking about this. How much does it actually take to infect the world with coronavirus? The extraordinary thing, one person's calculation was that it's about a Coke can of coronavirus has been made over the year, responsible for the most extraordinary disruption of life and so on. I think you thought it was about 2.5 kilos, I think, when you did your estimation.
Well, it kind of, the amount, the actual load, it all gets very complicated. There's also variables that are sort of, orders of magnitude, sort of, plus or minus, sort of many thousands in each direction. So that it actually starts to be quite a wide range, and you end up with everything from sort of a teaspoon up to sort of, 3 kilos of virus. So I kind of, when I did the calculation, I reckoned it was about 250 grams, which is about a Coke can. That's like a small bottle of water, I reckoned.
It's pretty amazing. It's actually a good point. If I share my screen here, and I'm not very good at this, I'm afraid. If I go to screen 2 here, which I hope you can now see, this is a picture that's been taken at Diamond in one of these amazing microscopes that they have there. And it's what you can actually see. This is inside cells, and you can see on it coronaviruses which are actually being built. You can see the spike proteins around there. You can see the virus, a new virus, and these are viruses which have been made by a patient or in a cell, which will then eventually erupt out of the cell. Now I need to get rid of that. How do I do that again? Get rid of stop share. And the person who was going to tell us about this amazing work at the Diamond facility, not far from where I was born and just the other side of Wantage, is David Owen. David, please introduce yourself.
Hi, so I'm a microscope scientist at Diamond Light Source. And normally I would introduce myself as someone who looks at small things, but after Marty Jopson, I guess I'm someone who looks at even smaller things. So we have, if I just want to share this. So we are like the particle accelerator. We accelerate electrons around a big donut, and we can use them to make X-rays, which allows us to look at really the fine structure of things. And then we also have electron microscopes. So this is so we've moved down a level of or upper level of magnification, and we're now looking at sort of single or tens of nanometers. And we then start to find that, as Marty was saying, we get radiation damage, so we have to take lots of individual pictures and put them together. And then hopefully we can then combine these, and we can start to look at the three-dimensional structures of these individual proteins. So these are sort of what we're calling the building blocks of the virus. And we can also do that with individual cells. And then what's sort of fun about this is we start getting a three-dimensional volume. We can look at the inside of a cell, and we can move through it, and we can really look for all of the different bits of havoc that the virus is causing by projecting all of these two-dimensional images together, and then we get this 3D view. So this is what we call a tomogram, and this is actually where one of those images that Roland just shared, and this is a factory where the virus is being assembled. So it's entered the cell, it's released its genetic material, and then it starts to produce all of its proteins. And what I like in the bottom right corner here, you see a mitochondria appear, and it's nice to have something that, like in the biology textbooks, you sort of think this is just a complete simplification, but actually that's just what it looks like.
And so those the thickish lines, those are the cell walls, are they there, David?
So in this case, we're looking at double membrane vesicles. So we are within a cell.
Inside a cell.
So this is the virus has entered the cell through the interaction of the spike protein and the ACE2 receptor. It sort of made this hole, it's punched itself in, and then the proteins that are on the surface of the virus cause the formation of these sort of bags, the little bags of chaos inside the cell, in which the virus can then start to replicate itself. And when we see these pictures, as you showed, of the virus, you see this corona. You have the spike proteins on the outside, and inside is just the bag of genetic material. But once this RNA, once this genetic material is actually seen by the cell, the virus can start to produce all of its own proteins. So here we've sort of got the ribosomes going along the virus gene, or the genome.
This is that strand there that's the RNA, that's the genetic part of the virus, and that's why it's getting into our cells, is to start using the machinery we have in our cells, which are those blobs, to make new proteins, which then make the new virus. Yeah,
and something that's interesting, and like I say, when we make our own proteins, so this blob in the middle is a ribosome, and that makes proteins in our cells, and we tend to make proteins one at a time, and they go off and do their job, various bits of chemistry and interesting reactions. Whereas the virus, it has this single strand of RNA, so it makes a single strand of protein, which then has to be chopped up.
And this is going on all the time inside an infected patient.
These cells are just being absolutely taken over. Our cells just become manufacturing sites to produce more virus. But what we've got going on here is, so now we're looking, so each one of these little blobs is probably 8 nanometers across. And this is this viral protease, this little heart-shaped protein that's sort of going along. It's chopping up this long strand of protein into the individual virus components that can then sort of help with assembly and help form more.
It's a bit like an Airfix kit comes with everything on one piece, and you have to get the pieces out before they can do their glue together.
And this is essential. if the virus cannot chop up its sort of long stranded protein, then it can't make any more. The life cycle cannot continue. So this gives us sort of an interesting opportunity to try and interfere with that process. So at the moment, we're looking at an X-ray crystal structure. So instead of using electrons, we're using X-rays. And this allows us to get to even higher resolution. So here we can look at the structure of things on the single nanometer or the 10th of the nanometer, the angstrom scale. And what's important about this scale is we can start to look at individual chemicals. So these are all little fragments of drugs. So we take, because drugs are normally quite large molecules with lots of features. So we chop them up, and we mix them with the protease itself, and then we see what sticks. And then we solve the high resolution structure of the protease with one of these little fragments of a drug. And then we hope that if we solve the structure of lots of different fragments, we can find different pieces of chemistry that bind to different bits of the protein, and we can find sort of ways of interfering with its with its sort of function.
And so this is what you've been doing at Diamond really for the past year, is trying to find the kinds of molecules that are still being invented, I guess, in the laboratories around the country and around the world, that will get into this particular protein. By stopping that one working, you then stop anything working. The virus might get into someone's cell, but it won't be able to replicate. It won't be able to make its copies of itself, and so you bring the whole thing to a halt.
So what we sort of did at the start of last year, sort of February and March, when this was first kicking off, is we get our first structure and we start having quite structures, very simple molecules that they stop the protease working, but they're not very good at it. You would need to sort of pump someone with grams and grams of it to have any effect. But as we release this information to chemists all over the world and they start combining these molecules into bigger, more complex molecules, then we end up with things that inhibit the protease sort of more and more effectively.
And there are other targets, are there? I mean, you can look at the protease molecule. I mean, I guess a lot of people are looking at the spike protein when it comes to vaccines.
Yeah, a lot of people are looking at the spike protein. People are looking at the viruses. They're fairly simple systems. They don't make a lot of proteins. If a virus that has a smaller genome as the SARS coronavirus makes a protein, it's likely essential. So people are targeting all of the different components of the SARS coronavirus to try and find different molecules that might work. And I think it is important that we do look at drugs and sort of therapeutics that are not necessarily vaccines, because there are always going to be people who might not be able to take the vaccine, or we might suddenly find that because of some mutation in the spike protein, the vaccine that we hoped was going to work isn't quite as effective as we would have liked. So we need to try as many different tactics as possible.
I'm curious, David, could you have done this kind of work? Well, I'm sure you couldn't have done it 10 years ago, but 5 years ago. I mean, it's one of the things I'm really interested in, is how the past few years of research has really brought things forwards.
What's changed is we're just better at it. We are faster and we are more efficient. X-ray crystallography, the physics of it was worked out about 100 years ago. 10 years ago when I started, I could solve the structures of proteins, but to be able to generate the amount of data at the rate that you need to try and make developments on a timescale that is actually useful, that's really quite recent. To sort of, if I take an individual protease, I can generate a structure a minute. So if you just sort of keep giving me different proteins with different drugs bound to them, I can just pump that information sort of onto the internet, onto the protein data bank as quickly as the sample can be produced. And that's been this huge change, because the more data we have, the more that we can look for patterns and we can see interesting features. And that's been this really nice thing about it's difficult to say nice thing about working on the coronavirus, but for once we've got all of the scientists effectively in the world pushing in the same direction. It's been so much more collaborative, and people have been releasing their information so much more quickly that we've been able to. I've done more exciting science in the last year than I've done in my entire career to date, and that's just because everyone is really helping each other to try and move the science fast and as efficiently as possible, instead of worrying about publication or who gets on what order on a author list.
I mean, I don't know how many drugs and proteins you've looked at, but have you? Do you have any sense of whether any of these are actually going to make it into the pharmacy somehow?
So something like we now have drugs that are sort of effective on the nanomolar range, so that is, we can dilute the drug, or we can give someone a small enough dose that it actually would make sense and it would have some effectiveness. And we're looking at repurposing of drugs, and I'm sure that'll be spoken about later, but looking for stuff that's already passed through clinical trials, whether it's some anti-cancer treatment, or so that at least if we find something that works, it can move fairly quickly. Hopefully it looks like they're just not going to be necessary. Like, I think the vaccines are going to have this thing sorted. But yeah, there's stuff coming through the pipeline, I think, good.
And if other coronaviruses, for example, were to emerge the way this one did, you would have a starting point.
Yeah.
To build on next time.
Yeah, I don't think this is going to be the last time we're thinking about coronavirus.
If you could just hold on there a bit, I'd like to bring in Verity. Verity Hill, if you unmute yourself there, Verity. Now you're representing another part of this amazing effort, which is there's been so much talk about variants recently, and variants are the kind of thing that you're finding with genetic techniques. Want to introduce yourself first?
Yes. Hello. My name is Verity. I'm a PhD researcher at the University of Edinburgh, and for the last year or so, like a lot of scientists, I think, I have suspended my work previously, which was on Ebola, and I've been working on analyzing genomes for, it's called COG-UK, and it's a UK-wide sequencing consortium. So what happens is that kind of across the country, there are lots of centers for sequencing that kind of already existed. There's a lot of sequencing expertise within the UK. And samples come from NHS trusts, care homes, the population, anywhere. They're tested in things like the Lighthouse labs, and then the samples are sent to one of our sequencing centers. They are sequenced, and then the data is put onto this one central server. It's cleaned, it's analyzed, and then it's made public. So we've been tracking variants since kind of the first one we discovered first one was discovered, or was flagged maybe more accurately, in Kent in kind of December time, which we call B.1.1.7. This is a lineage naming system that we came up with about a year ago. It's a bit of a mouthful, but it gets away from calling it like the UK variant or the South Africa variant, which we try not to do. So we were worried about that because it had a mutation in it that was a little bit concerning. We thought it might lead to some increased transmissibility. And we have been able to track that thanks to a combination of all of the sequencing in the UK, but also there's been, I think as David was kind of talking about for the proteins, there's all this amazing global effort for this as well. So we've got like, I think there's like almost 500,000 SARS-CoV-2 genomes from across the world, and countries that haven't done much sequencing before have been able to upload sequences and contribute to this global effort. And it's really an amazing global thing. So we've been able to track various different variants. So we've got the one that was found in the UK. We've got one called B.1.351, which was first found in South Africa. And we've got one that we call P.1, which was first found in Brazil. And the one first found in South Africa and the one first found in Brazil are the ones we're worried about for what's called immune escape, because sorry?
I was going to say we'll come to that. We'll come to the details of these in a moment, if that's right. I mean, what's interesting is that obviously people have been very worried about these recent developments in the virus because they seem to make it move faster, for example, or it can escape some of the vaccines, maybe. But you've actually been following them right from the beginning, as I understand it, because in a sense that very small mutations that have no effect are nevertheless a kind of a fingerprint for you. I think you showed me earlier, you've got a quite nice picture where you were showing how all the different variants were sort of arriving a year ago. So, you know, we now know that most of the cases that came into the UK did not come from China, but came from other parts of the world.
Yeah, exactly. So actually, usually what the methods that I use in the kind of analysis I do, I'm usually not at all interested in what the mutations actually do to the virus. I'm usually only interested in the fact that there are mutations, exactly in the way that you're describing, that it's like a type of fingerprint. So using the genomes taken in the UK and across the world, and combining it with some really clever maths that an epidemiologist at Oxford did, we were able to estimate where we thought the virus had come from into the UK. And it was mostly, there were a few lineages that we think came from China, but actually mostly it seemed to come from the rest of Europe. Spain was quite prevalent in the analysis we did, just because the UK and Spain have very strong links, and like a lot of travel between the UK and Spain. And it's almost certain that, as all of these things were coming into the UK was probably, spitting them straight back out at the same places. So this is in no way, of course, to ascribe any kind of blame. It's just useful to know where things come from. And that's only possible because everybody is sequencing and sharing.
Can you pull that one up on your screen?
Yeah, sure. I was thinking it might be, here we go. Yeah, I'll make that. Yeah, so this is the figure that I was talking about. This is from a paper that came out actually quite recently. I think it came out in January in the end.
But I just find it extraordinary. It's such detective work that you can, you can look at these little details in the genome, and you're scanning what, every 1 in 10 genomes of any positive patient, something like that?
I'm not sure about the exact percentages. It's not very many though, but that's the joy of genomic data and why it's so useful as a supplement to traditional epidemiological techniques. Because with traditional epidemiology, you kind of have to like find the person that moved from country A to country B, right? Or somebody who is infected by them, and you have to interview them and you have to find that out. And kind of this is very kind of traditional. Like, there's a lot more maths involved now, but what's really cool about genomes is that you have this whole big family tree of viruses, and you can infer all sorts of events that have happened quite far back in the tree that we haven't sampled directly. So usually I work on Ebola, and for the West African Ebola outbreak, they sequenced about 5% of cases, and we can still get a lot of information about the dynamics of that epidemic just from the genomes, and with a little bit of extra data, like the date the sequence was taken and where the sequence was taken.
Yeah.
So yeah,
I mean, but it's just amazing. So you can see in this graph that around just before the 16th of February, you're seeing these cases coming in from Italy. Probably people have been on skiing holidays or something. Then I guess are they from Spain, the really big birds? But it's really interesting. There were multiple times that the virus came into this country at the beginning of the pandemic, and you can see that.
Well over 1,000 times, probably.
1,000 times.
Yeah, and like obviously our estimates are not, they won't be exact. There are error bars on these, but it was at least 1,000, we think.
And you did. It was the same process when after the summer holidays in Spain, when case numbers were quite low, people were bringing a different variant back from Spain, and again, you could see that, I think, across the whole of Europe.
Yeah. There was one lineage called B.1.177.
We're not keeping track of them.
I know. It just so happens that a lot of the ones we're interested in are 1s and 7s. It's just bad luck. But yeah, that one actually that spread, and that took over quite a lot of Europe. And that we don't think it actually had any kind of biological advantage. It was just coming into a point where a lot of countries had relaxed restrictions and that kind of thing. But yeah, and there's analyses like this going on at the moment, trying to work out the relative impact of, say, like virus transmission chains in the UK that were sort of bubbling along at a fairly low level, but weren't stamped out, versus new introductions from other parts of the world, trying to work out the relative importance of those two things in driving the second wave. So that's analysis that's happening at the moment. It's quite a complicated question. It turns out it's very hard to tease apart these factors, but we're trying.
And so one of the reasons you were able to do this was because, I guess, with the Human Genome Project, the Sanger Centre near Cambridge, there's a huge amount of genomic work going on in the UK that you could already build on. I think you've also got with you, though, a piece of kit which has totally transformed the way that genetics is done. Introduce us to it.
Yeah, okay, so this is a Minion, and you can see how really small. I mean, I have quite small hands, but it's really small anyway, I promise. So this is called a Minion. It's made by Oxford Nanopore, and what it does is it opens like this, and then in here you put what's called a flow cell. So I think this is a display one, so I can't remember if the flow cell is like a little bit different to this, but you pipette your sample into this flow cell, and you can run lots and lots of samples at one time. And there are tiny, tiny pores in this, or nanopores in this. And what happens is the DNA or the RNA gets pulled through these pores, and the way that the pore deforms with each letter of DNA is measured electrically. And that electrical signal from the deformation of the pore is then interpreted by a computer, and it tells you what DNA or RNA letter.
So that RNA thread, which David was showing us earlier, running through the ribosome, this molecular machine, you're basically doing the same thing, running threads of RNA through, or DNA perhaps, I don't know, but through that thing, and it's just reading out the letter by letter. Yeah. It's tiny compared to the machines that they were even using 5 years ago.
Yeah, and it's really cool because it will run off a laptop as well. So you don't. I
thought I saw a USB thing. Did I see a USB connection at the end?
It's not quite a USB.
Oh right, it's a bigger.
Yeah, but you
easily fooled.
But you do run it. You can run it off a laptop, and that's really, really cool because in a lot of places, consistent power is not something you can rely on, or at least without blips going to a generator or something like that. So while it won't run for a really long time on a laptop, it can tide you over until your power comes back on and you don't lose all your data. With traditional big sequencing machines, if the power goes off, like that can be just it. You can just lose everything. And especially if you're talking about samples that are like blood from somebody that's really sick, or I can't remember what they're called, the kind of encouraged sputum samples. Where you have to really make someone cough. They're not nice for the patient, right? So you don't want to be losing your samples. So that this can be powered off a laptop is also very, very useful. And it's part of the there are some collaborators of ours in Cambridge that have been working on the lab in a suitcase thing. So they put one of these, and it's basically all the equipment you need to go from patient sample to sequence. All fits in. I think it might be like 2 big suitcases, but you can take it on a plane, and then you can take it wherever you need to go. Includes like car batteries to power stuff, and like they have to be very inventive. They've got like tents that you can use UV on, so it will kill things. So this has been used for Ebola, which is like the highest like containment category of virus, and you can still use this equipment for that.
And that was the first time I heard it. It's amazing stuff. Very, very briefly, Verity, you're pretty much at the beginning of your scientific career. I mean, what's it been? David said how important it's been to him
Yeah.
For you, what was it like suddenly to be involved in this?
Oh, well, I mean, so I was 3 years into my PhD, and about that time, everybody, I think, starts to get very disillusioned with what they're doing. I was like, oh, I'm not sure.
David's smiling there.
Yeah, it's like, oh, this is fine, but I don't think anybody really cares about what I'm doing. And I've got very down into one very narrow part of one very narrow piece of science. And then this started happening, and suddenly everyone cared what I was doing. And yeah, it was definitely very motivating. And I have got to do some really cool science. It's exactly what David was saying. Things have come on in leaps and bounds. We've never had this many genomes before. There's 500,000 genomes, and 200,000 at least of those are from the UK. So we're dealing with data sets 100 times bigger than we've had before. So we've had to come up with all of these new methods of how to deal with that enormous data set. And
you've been overwhelmed.
Yeah, but it's been scientifically very fulfilling, if completely bizarre.
Yeah. Well, that's brilliant. So this is when I was hoping to talk to Nisreen Alwan. I think, Nisreen, we've been talking here about treatments, about understanding the virus. Obviously, there's been this enormous focus. Sorry, there's a stupid noise going on in the background here. That's useful. Nisreen, introduce yourself, and then that'll shut my microphone down.
Yeah, sure. Thank you, Roland. I'm Dr. Nisreen Alwan. I'm an associate professor in public health at the University of Southampton. I normally, before the pandemic, my specialty is public health and epidemiology, and it was focused on maternal and child health. And with the pandemic, like everybody else, life has changed. We focus a lot on COVID-19, public health response, but also I got COVID-19 symptoms back in March, and I was one of these unlucky people not so unlucky, I didn't have to go to hospital. It wasn't really severe at all, but it didn't completely go away, and it kept coming and relapsing. So what is now called long COVID. So I then been spending the last few months trying to raise awareness about long COVID, talking about it, and really trying to shift a bit the narrative, in addition to doing research on it, but trying to largely shift the narrative of looking at coronavirus as just, black and white, something that kills a minority of people and doesn't touch the rest, because there is evidence now more and more that there's, a significant amount of people are affected by long COVID, which is basically them getting COVID, but then not going back to their previous health, and to varying degrees, some of them very debilitated by it. And some people like me really who've kind of tried to adapt their lives to these symptoms, but they're not pleasant, and they're quite worrying, because you don't know what's happening, what's going on, whether they'll completely go away.
So, describe your own symptoms first.
Yeah, so I had, so in March, we were told cough and fever, and somebody with my age group and my profile wouldn't, it would be like a flu-like illness of anything. And that's what I had. But I had a wider range of symptoms, which we now know are a feature of, coronavirus infection. And we didn't so much at the time, So I had the fever and the cough, but I had this feeling of chest heaviness. I had the muscle aches, I had the chills. But I also had some, abdominal symptoms, so abdominal pain and diarrhea, which we all now are, now we know they're features of coronavirus. And then I sort of kind of got better. It took much longer, week 2. By week 3, I was really worried, because nobody was saying it would last that long in people who weren't hospitalized. But people were talking about it, and there were posts on social media of people I know saying, what's going on? I'm not, I feel like I'm getting better, and the next day, the symptoms are coming back, which was what I was experiencing. And then it just, the following months is kind of a cycle of this, where you feel like you're on the mend, you try and get your, normal activity levels, and your life back, and then symptoms hit you. And with time, you start recognizing what brings them on, and you try and avoid doing things that would bring them on, but sometimes you can't completely avoid it. Some
What,
people,
what kind of prevalence is there for long COVID? How many, what percentage of people do you think are suffering from it?
so this has been the million-dollar question. Is for many months people weren't measuring at all. Now that we've got some statistics coming, and they vary really, but the ONS, the Office of National Statistics, estimate is 1 in 10 will have persisting symptoms 12 weeks after the infection. But then there's no breakdown of severity of these symptoms, and this is based on a random sample of the population. We've got many more figures based on hospitalized people, and we know it's very common who people go to ICU and hospital to not get their full health back for months after. But what's worrying is that it seems the long COVID is not correlated to the severity of infection at the start. So many of the people who had mild illness like me have it as well. So that's an estimate of 110. Many other studies emerging from European countries, from the US, which say actually it could be more than that, could be up to 1 in 4, for example, people, with the fatigue being the most common symptom, but also have those other selection of symptoms. One thing is worrying, which is emerging in people, is the cognitive symptoms, which is poor memory, concentration, what people are describing as brain fog.
Yeah, heard that phrase.
And it seems to be more, they develop, in some people they develop, not from the start, they're not present from the start, or they get worse with time. And that's a big mystery, particularly with these cognitive symptoms.
And what is anything known about the causes? I guess the key one is it the virus itself, or as I understand it, an awful lot. Martin Landry has just joined us. And the other aspect that I keep on hearing about the coronavirus is the immune response is actually responsible for an awful lot of the problems from it.
Yeah. So absolutely. Well, the long COVID is really an umbrella thing. I mean, it could be we're having different conditions under this big umbrella, and so it could be the immune system. So we've got examples of other diseases where there's an autoimmune response. So the immune system goes into overdrive, and it could, some of these diseases have cyclical nature, like lupus, for example, or multiple sclerosis. But also, I think we're not at a stage that we can rule out persistent virus. There have been some studies to say that there are persistent virus in biopsies from the gut, for example, for many months, so we can't rule this out. And also, the other mechanism is basically just damage to tissues that happen from the infection. So some scarring to tissues. But and that could be another mechanism. And it could be we have more than one mechanism in people, and we have different conditions under the long COVID spectrum, because it's such a wide clinical spectrum.
So it's one of the things I've realized as this pandemic's gone on is so many of these questions are so difficult to answer, because you can't just, I think in the public, we think we can just click our fingers and there we have the answers. But since Martin's joined us, I gather you had some problems. We sent you the wrong link, I think, Martin. I'm so sorry. Could we just talk a bit about the inflammation, because you run the drugs trials system called Recovery, which is, I think, been the outstanding success in terms of drug discovery, treatment discovery, I should say, in this. And you had yet another success just last week with, now I'm going to practice, I've been practicing tocilizumab.
Oh, very good, very good. Yes, you better than the Prime Minister. Yeah, Tossi's good friend, I think. Yeah. I mean, this time last year, COVID was an entirely new disease. We had little knowledge of its nature. Certainly, it's not its clinical course, and we had zero knowledge of how to treat it. And hundreds of drugs were proposed from all sorts of sources. And the question was, how do you filter through all that list and try and find the ones that are going to work and the ones that are not? And it's not always obvious. And there's some great basic science, some great structural biology and so on. But ultimately, it comes down to clinically, here's a drug we think might work, we hope will work, but do we know that it actually works? And that was where the trials came in. And the recovery trial has now 37,000 NHS patients have taken part in the trial. That's hospital inpatients. And from that, we've learned a lot. And one of the things we've learned is that actually Yes, there's a problem with the virus, which I can still see on the screen there, and it's beautiful pictures. Yes, there's a problem with the virus, but the equal and possibly even the greater problem is how we respond to that virus. Initially, the immune system is our friend. It tries to attack that virus, destroy it, prevent it entering cells, prevent it replicating and causing damage. But over time, perhaps towards the end of the first week, as some people get sicker, the immune system is no longer our friend and starts to actually drive much of the inflammation in the lungs and a lot of the damage we see.
And,
so on the trials,
and that seems to be as I understand it, the two drugs that you found that really do the job are the ones that tackle that over response.
Absolutely. I mean, whether tackling the virus earlier in disease would be helpful as well, we don't know yet. But what we do know is amongst those sicker patients, sick enough to go into hospital, to need oxygen, that some of them end up needing to have ventilators, whether that's a face mask ventilator or if you like, an old-fashioned intensive ventilator. In those patients, then if you suppress the immune system, and there are two drugs we know work. One is with dexamethasone, which is a steroid drug been around for decades. Then you will improve the chances of survival and recovery. And the second one, which we identified a week ago, was this drug tocilizumab, an arthritis drug targeted at one very specific part of the immune system. Blocking that one specific part produces some additional benefits. So yeah, the two winners so far have been about how we deal with the response to the virus, as opposed to how we do with the virus itself.
And there was at the beginning, there were, as you said, I've no idea how you sifted through the list when you got them, but I mean, one of the ones I think you dealt with was hydroxychloroquine, this malaria drug, which everyone was gung-ho for, cheap, easy, lots of anecdotal evidence that it was really helping out. And in a sense, I think that example proves why the very thoughtful way that the recovery trial could test it, ruled it out, said that's absolutely not any use.
Yeah, and we didn't know. It was a good hypothesis. There was some science, that's the first thing, that suggested that hydroxychloroquine might be a useful treatment. Secondly, there was some not terribly good science about how some of those early analyses and early clinical studies were done and reported. And thirdly, in the context of an exploding pandemic, threatening particularly as actually the developed world very drastically last spring, the US, France, UK, and then later on into Brazil and India and so on, then people were desperate for treatments. And so when somebody says, yes, we've got these treatments that's cheap, cheerful, widely available, then there becomes a clamor from clinicians, from the public, from politicians, and from all sorts of others. Well, it can't do any harm. Let's just use it. And our view was throwing drugs at patients in the hope that they might work without knowing that they work would waste resources. It was a false promise. And that what we needed to do was find out if it works, that's great. Let's give it to everybody. And if it doesn't work, that's useful knowledge. Let's give it to nobody. It turned out in the case of hydroxychloroquine, by the time patients get into hospital, it is completely and utterly useless.
I mean, it sounds like it sounds like a defeat, but in a sense, by meaning you're not wasting resources, it is a victory in its own right.
Yeah, so
in sort of 20 seconds.
Yeah, and another one, just to very briefly, is azithromycin. Azithromycin is an antibiotic. Lots of good reasons for thinking it might work. Do the trial, it turns out it doesn't work. We don't waste resources, but by goodness, we need antibiotics to be antibiotics. We don't want drug-resistant infection. So a very good way of saving resources. But the successes have been dexamethasone, tocilizumab, reducing inflammation, saving lives in the sickest patients. And we couldn't have done that without the clinical trials.
And I was hoping to get a bit of interchange between you and Nisreen, because I'm really interested in whether there's going to be any chance of extending these trials into something like long COVID, because even once we've cured the disease, it sounds like there will still be people who might need help. Absolutely.
Well, I mean, the first, and this is not to trivialize it, but the first challenge was with COVID was survival. This almost sounds like trivializing, and it's not, but you cannot get long COVID if you do not survive short COVID. So that was the number one challenge. And
okay, I'm told we're about to be cut off, so very quickly get my
the other challenge is, yes, there will be There need to be new treatments. We need to understand more about the disease to work out which bits of the disease, and it's probably several, we should be thinking about treating with which sorts of drugs.
Brill. Martin, thanks very much. Nisreen, thank you very much. Say bye-bye. We only see you when you say something.
Bye. Thank you very
David,
much.
thank you so much for talking to us. There was a question I didn't get to for you. Verity as well. Thank you so much. And Marty, I should say. Marty, have you got your virus there still to say goodbye to us? And just to say thanks to all of you, and also please fill in the form if you've been listening to this, and we'll get the feedback that will hopefully make this even better, or at least better next year. Thank you to everybody.